At the beginning of this chapter, organisms that have a single cell, that are composed of single cell, are known as unicellular, and they include amoeba, fungi, there is protoxta. Organisms that composed of many cells are known as multicellular, and they include animalia and the plantae. We go direct to the definition of the cell, and the cell is a basic unit of organism. The cell is a basic unit of organism, and the cells are too small to be seen with naked eyes. So we used to use special equipment that is known as a microscope. And there is two types of microscope: electron microscope and the light microscope. First, we start as light microscope. Light microscope is used to give finer details that handling cannot give, and it is called a light microscope because it uses light to illuminate the specimen. So, how can we find magnification? Magnification is equal to eyepiece magnification times objective lens magnification. Then from there, we'll go to electron microscope. Electron microscope, organism that can be seen with electron microscope, and we'll go direct to difference between electron microscope and the light microscope. Light microscope is used light to illuminate the specimen. Electron microscope is used a beam of electron to illuminate the specimen. Light microscope, it is used a glass lens for magnification. Electron microscope is used glass lens for electromagnetic for magnification. It has light microscope, it has a low resolving power. Electron microscope, it has a high resolving power. It has low magnification power. Electron microscope has a high magnification power. Light microscope, organism that can be specimen that can be seen with light microscope can be dead or living, but specimen that can be seen in electron microscope are always dead. Then from there we'll go to cell structure as seen under the light microscope, and cell structure that can be seen under the electron microscope. Then we'll go difference. Then we'll go direct to the difference between plant cell and animal cell. Plant cell is usually large. Animal cell is usually is often smaller. Plant cell has chloroplast, but animal cell have no chloroplast. Plant cell lacks centrioles, but animal cell have centrioles. Plant cell have cell wall, but animal cell do not have a cell wall. Then from there, we'll go direct to estimation of the cell size. Estimation of the cell size. As estimation of the cell size, we'll go to direct formula is equal to cell length is equal to diameter of the field of view over the number of the cell. Then from there we'll go to organelles that are organelles of the cell. Cell organelles. One we'll start with cell membrane. Cell membrane is also called plasmamembrane, and is composed of double layers, which are known as phospholipid layer. It is used to regulate the movement of material in and out of the cell. From there we go to mitochondria, and this is a site for respiration, and it is also used for energy, to yield energy. Then from there we'll go to cytoplasm. Cytoplasm is where all organelles will take place in cytoplasm. From there we'll go direct to endoplasmic reticulum. And endoplasmic reticulum is of two types, where there is rough endoplasmic reticulum and a smooth endoplasmic reticulum. And the rough endoplasmic reticulum is called rough endoplasmic reticulum because it has ribosomes, and this function of ribosome is to transport is a site for protein synthesis. Then rough endoplasmic reticulum will transport this protein. A smooth endoplasmic reticulum will transport lipids. Then from there we'll go to another organelle which is known as nucleolus, which is used for foundation of ribosomes. Nucleolus controls all activities of the cell. Then from there we'll go to another organelle which is known as vacuoles. Vacuoles is used for storage. Can store food, can store water. Then from there we'll go to cell specialization, tissues, organs, and organ system. We'll start with cell specialization, and we'll start with root hair cell. Root hair cell is used to increase the surface area for absorption of water and the mineral salts. Then from there we'll go to guard cells. Guard cell is used to control the opening and closing of stomata. From there we'll go to muscle cell. Muscle cell will have contractive fibrilles which will relax and bring in about the movement. Then from there we'll go to organs. Organ will go to organs. Organ is a distinct part of an organism